Bismillah ar-Rahman ar-Rahim. The title of our practical session is in renal module. It's ligand assay, and the ligand is due to use a substance that bind either with the antibody or antigen, and this is called the conjugate. And this substance that bind to the antibody either fluorescent material or enzyme or radioactive material. First, we have the immunofluorescence test, and it's called so due to use the conjugate, which is the fluorescent material, and it's of two types, direct and indirect method. The direct method, we use the tissue from the patient, like skin biopsy or renal biopsy, that contain the antigen in the skin or in the glomeruli of the kidney, and then we add to it the conjugate, which is the antibody labeled with fluorescent material, and then examined under the fluorescent microscope. And this is used to detect the autoimmune disease in the glomerulonephritis or in the pemphigus vulgaris, in the which is autoimmune disease of the skin, characterized by blisters filled with fluid. While the skin type of the immunofluorescence is indirect type, and we use the biopsy from the animal, which is usually used the rat or mouse, and we took biopsy from the liver, from the stomach, from the esophagus, and then we add to it the patient serum that contain the antibodies, and then we add the conjugate, which is the antibody labeled with the fluorescent material. And this indirect method used to detect the autoimmune disease, like anti-nuclear autoantibodies, anti-smooth muscle autoantibodies. And here the immunofluorescence test, usually it's used the fluorescent microscope, not ordinary microscope, and examine the slide, and then we see the apple green color of the tissue, and this is used to detect the autoimmune disease of the thyroid, and this is the autoantibodies against the thyroglobulin, and here against the thyroid cell. The second type of the ligand assay is ELISA, which is abbreviation of enzyme-linked immunosorbent assay. And here we use the microtiter plate, and this plate will be coated with the antigen, and from the company that synthesized the kit, and then we add the patient serum that contains the antibody, and then we do washing to get rid from the excess antibodies, and then we add the ligand, which is the antibody labeled with the enzyme, and then wash, and then add the chromogen and substrate, and then we get a color which is yellow color, and this is used to detect the antibodies against the HIV, antibodies against the hepatitis B surface antigen. And the principle of the ELISA, the antibody labeled with enzyme, usually use the horseradish peroxidase enzyme or alkaline phosphatase enzyme, and then add the substrate, which is the H2O2, and this leads to liberation of the oxygen, and the oxygen will oxidize the reduced chromogen, and then we end with oxidized chromogen and water. And this is H2O, not H2O2, water, H2O. And here the plate of the microtiter plate of the ELISA, and each well, which is for one patient, and read the color by spectrophotometer or ELISA reader, and we get the concentration of the antibody, and it is the quantitative method give the concentration of the antibodies in the serum. The another test, which is a RIA, which is abbreviation of the radioimmunoassay, and here we use radioactive material instead of enzyme. It's the same principle as ELISA and previously used for the hormonal assay. Nowadays no longer used because of its difficulty to get rid from the radioactive materials that used in the test. And the another new test, which is immunochromatography, which is a qualitative and rapid test using a dipstick membrane coated with monoclonal antibodies against the antigens. And here the conjugate, which is a protein A colloidal gold conjugate, and using in many tests like detection of malarial antigen and the pregnancy test, and nowadays used to detection COVID-19 virus by detection the antibodies against this COVID-19 antigens. And it is a rapid test within few minutes, and it's used to early detection of the infection. And it's used either to detect the IgG, which is for the secondary immune response, or IgM, which is for the primary immune response that indicate recent infection, or the detection the IgG that indicate past infection. And here the immunochromatography, which is used for the pregnancy and nowadays for the COVID-19, and used for the malaria, used for the process-specific antigen, used for the viral infection. And this is very easy and done, and we got our results within minutes. Then another test, which is old test, and it's used or also for detection antigen or antibody, and it's called complement fixation test. And here we have two tubes. Here we add the antigen, and here we have the antigen, and then add the patient serum to each tube. If the patient is infected, so will develop antibodies against these antigen. If it's not infected, so there is no antibody against these antigens. So this antigen antibody immune complex, you cannot see this reaction. So add to it the complement, and this leads to antigen antibody complement fixation, while here the complement will remain free. And then we add the indicator cell, which is called receptor cell, and to see the reaction between the antigen antibody and the complement. And here, when the complement consumed by this antigen antibody immune complex, so the receptor or indicator cells or sensitized RBC will remain free. While here, when there is no antibodies, the antigen and antibody will remain, the antigen and the complement remain free, and the complement will bind to sensitized cell, lead to destruction of the RBC and release the hemoglobin. And the indicator cell or receptor, which is consist from the RBC coated with the antibodies. So when there is rupture of the RBC, indicate that the patient negative and no had infection. And when there is no rupture to the RBC, indicates that the patient is infected and develop antibodies against the antigens. Now we reach to the end of the humoral immune response. Now we have a new subject, which is cellular immunoassay. To 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 do the cellular immunity and to detect the cellular status of the immune system. First, we must separate the lymphocyte. And here we use the media, which is called lymphocyte separation media, or called the Ficol HIPEC media. And it had specific gravity 1.00078. And we add the blood from the patients or any individual on this media, and then we did centrifugation, so we'll get 4 layers: plasma and platelets, mononuclear cells and lymphocytes, and the third layer lymphocyte separation media at the bottom, RBCs and granulocytes. So here, this is the platelets with the 4th layer. Here is the plasma only, no platelets, mononuclear cells, lymphocyte separation media, RBC, granulocytes, and the platelets. And here the Ficol at the bottom of the test tube, and add to it diluted defibrinated blood, and then centrifugation, and then we'll separate into 4 layers: the RBC, granulocytes, platelets at the bottom, and then the Ficol, and then the lymphocyte, and then diluted plasma. We will take the lymphocyte because cellular immune response, in order to assess its function, we need lymphocyte. The lymphocyte first, enumeration of T-lymphocyte, and this can be achieved using the sheep RBCs because T-lymphocyte has a receptor CD2 receptor that bind to the sheep RBC, lead to formation rosette. And we calculate the percentage of rosette cells using hemocytometer jumper, hemocytometer counter, and then detect how many from 100 cells had or got rosetting, and get the percentage. And this is the rosetting T cells. Second function, the enumeration of the B lymphocyte. As you know, the B lymphocyte had on its cell surface antibodies, and then we add to it the conjugate, and this is the direct immunofluorescence, and then examine under the fluorescent microscope, and we see the cell wall of the B lymphocyte will be apple green and fluoresce. This indicates B cell, while if it is not B cell, it's remain red in its color. And also, we calculate 100% cells and how many of them had fluorescence cell wall get percentage. The third test is enumeration of T lymphocyte subset CD4 and CD8 using indirect immunofluorescence test, and this is the T cytotoxic, and as you know, on its cell surface presents a marker CD8, and we add to it monoclonal antibodies against the CD8, and then we add the conjugate, which consists from the antibodies coupled with the fluorescent material, and also will give us the fluorescent apple green fluorescent on the cell surface of the T lymphocyte and examine how many cells that had fluorescent cell wall and calculate the percentage. The last function is using lymphocyte in tissue typing, whether serological method or molecular method, as mentioned in the lecture, sequence-specific primer. And in serological method, which is Terasaki method or HLA typing, here we have the lymphocyte, and on its cell surface, as you know, has the HLA molecules. Here, for example, the HLA B8. And add to it monoclonal antibodies against this B8, and this is the antigen antibody immune complex. Then we add the complement that lead to fixation of this reaction and lead to activation. The classical pathway of the complement lead to membrane attack complex MAC and lead to lysis and rupture of the T or any lymphocyte. And then we add the stain, which is a trypan blue. When there is rupture of the cell wall, this stain will enter the cell and will have blue color, while live one did not have any color because it is live and there is no destruction of the cell wall. When there is a destruction, so this means that the patients had B8. And another method, the molecular method, and we extract the DNA using a sequence-specific primer, extract DNA from the lymphocyte, and the DNA, which is the double-stranded DNA, and then we add to it nucleotide, tag polymerase, and the primer of the specific HLA allele, and put them in the PCR, and the PCR change the temperature starting from 95 centigrade, lead to denaturation of the double-stranded DNA into single-stranded DNA, and then the machine will decrease the temperature to 65 centigrade, and this lead to annealing of the primer if it is compatible with the DNA, annealing with the primer. And then the machine PCR increase the temperature to 72 centigrade, and here lead to extension of the DNA by the nucleotide. And here we end with 2 copies, and this process will be repeated 30 times. This PCR machine repeated this 30 times. So we start with single copy of double-stranded DNA and end with multiple copies of the double-stranded DNA. And then we'll take to it gel electrophoresis, and here we find the migration of the bond that had proliferation, while it is not compatible with the allele, so there is no migration of the DNA bond. And we have a map of each allele that's put in this line, and we will get the HLA typing of the patients or of the individual. And thank you.